Hello everyone, my name is Alison DeVault-Enck and I work on specialized sequencing solutions at Dicel Arbor Biosciences. Our company enables researchers to tackle complex questions with our products and services that are built for specialized genomics. Today I'll be sharing insights from a recent Nature Plants paper that redefines species boundaries in Liberica coffee, which is a crop with a huge potential for climate resilience. I'll also show you how our myBaits Angiosperms-353 kit played a pivotal role in this breakthrough. The specific study I'll be highlighting today is this recent publication from August 2025 in Nature Plants from Davis et al. The authors of this research paper come from UK and African research institutes, the Royal Botanic Gardens Kew in the UK, the National Agriculture Research Organization in Uganda, Makerere University Herbarium in Uganda, and the National Herbarium of Cameroon. This paper is available open access with this DOI, and I highly encourage you to check it out as soon as you can. Let's start with the challenge that these researchers faced. So coffee is one of the most traded commodities globally. It's valued over $200 billion for the global market, and it supports millions of livelihoods along the coffee supply chain. But climate change is putting this entire system at risk. Arabica and Robusta are two of the dominant commercial coffee species. They are increasingly vulnerable to the impacts of climate change, such as rising temperatures, pests, and changing rainfall. But there are actually many coffee species. So a minor global commercial species, Liberica coffee, has long been considered a single species with two varieties, known as Liberica and Excelsa. Farmers and breeders have been intrigued because it has many resilient traits that could be relevant, such as drought tolerance and disease resistance, but there has not been a full understanding of its taxonomy, somewhat controversial. So why does this matter? If researchers don't know whether they're dealing with one species or multiple species and what population structures actually look like, it's very difficult for breeding programs to effectively harness genetic diversity to improve species. And conservation efforts also suffer because it's difficult to prioritize which populations to protect. So the challenges here is clear. There's a need to resolve species boundaries in Liberica coffee using robust genomic evidence in order to enable informed breeding and conservation decisions. And that is where our technology came into the picture. To tackle this high-resolution phylogenomics challenge, the researchers utilized Dicel Arbor's myBaits Angiosperms-353 kit. This kit, this target capture panel targets 353 nuclear genes, which are conserved across flowering plants, or angiosperms. It's a universal panel which works across an enormous diversity of species and taxonomic breadth, making it perfect for comparative studies. It's highly flexible. The myBaits technology can handle even degraded DNA from herbarium specimens, which is critical for studies like this that need to integrate historical samples. And it's standardized. It provides a common set of loci for reproducible analysis. So in brief, this technology works basically by increasing the efficiency of next generation sequencing, or NGS, by focusing on informative genomic regions. So the DNA is extracted from a biological sample. It's prepared into a barcoded NGS library, enriched with the myBaits kit prior to sequencing and analysis. This specific panel, Angiosperms-353, was originally published by Johnson, Pokorny, Dodsworth et al. In Systematic Biology back in 2019. Since then, it's been used in hundreds of studies on angiosperms that have helped to resolve plant tree of life, perform phylogenetics, as well as even higher resolution applications such as population genetics. I'm highlighting here just a very small sample of papers from the last year or so, which demonstrate the very broad use of Angiosperms-353 across a wide variety of diverse plant groups, including many agriculturally important applications. But to address their specific research questions about Liberica coffee species delineations, Davis et al. Extracted DNA from 55 accessions of Liberica coffee, including wild and historical wild populations and historical collections. Some of the samples that they included in their study were over 100 years old. They enriched those samples with the myBaits Angiosperms-353 panel and sequenced those on Illumina. In addition, they performed detailed morphological analyses on over 700 living and herbarium collection specimens. And they were able to perform robust phylogenetic analyses by combining genetic, morphological, and geographic information, which allowed them to rebuild this robust phylogeny, something that traditional markers like ITS or plasmids alone couldn't achieve with that same resolution. So what did they discover? The results were transformative for our understanding of Liberica species identification. Instead of just one species, their data make it clear that Liberica comprises 3 distinct species, which are called Coffea liberica, Coffea de Wevre, which is the Excelsa group, and Coffea clanae. In this phylogenetic tree from their research, de Wevre is here in green as a distinct group, clanae and Liberica with other coffee species shown here in gray. These are 3 distinct monophyletic species, which brings, considering these 3 new species along with the other Coffea species in the genus, this brings the total number of overall known Coffea species to 133. The new species delineations are fully supported by both the genetic as well as morphological and geographic information that the researchers analyzed. So why does this matter? It's not just a so why does this matter? The 3 species have geographically distinct wild distributions in Africa, and the natural range within and between those species is very diverse. This is basically these large natural ranges with a lot of diversity in ecology and geography means that there's a lot of local population adaptations that allow them to occupy unique ecological niches in Africa. So for example, Liberica thrives in lowland forests. De Wevre has a lot of drought tolerance and also tolerates poor soil. And clanae is adapted to specific microclimates in Central Africa. Again, this isn't just an academic exercise. It really is important for coffee crop improvement because this type of research opens new doors for breeding climate resilient coffee varieties by increasing our understanding of genetic diversity and helping to guide conservation priorities. So for example, there are areas in the world where traditional Robusta coffee farming is suffering and because of climate stress, and these type of new developments could help farmers maintain their coffee crop yields in those places due to species like de Wevre, which have a lot of drought tolerance. And that's the kind of impact that having genomic clarity can help deliver. So what's the takeaways for us? In short, genomic tools like Angiosperms-353 are helping to revolutionize plant systematics and crop improvement. By resolving Liberica species boundaries, the researchers here unlocked new pathways for sustainable coffee production in a changing climate. So by integrating advanced high-resolution genomic technologies such as Angiosperms-353 into their agriculture research, this can help drive innovation, enhance crop resilience, and help support global food security, global farming security, and livelihood security. So if you happen to be working on phylogenetics, conservation genetics, or plant breeding, universal kits like Angiosperms-353 can help accelerate your research, handle challenging samples, and help you deliver reproducible, high-impact, cost-effective, next-generation sequencing results. Our team would love to discuss project ideas with you. Please reach out today. You can find our contact on our website here, arborbiosci.com, or reach us via email. I thank you very much for your attention, and especially to Davis et al. Researchers for their absolutely fascinating study, which unlocks new insights on coffee species genetics and contributes to the future of sustainable global crop production. Thank you again, and have a great day.